Good morning, everybody. We've got Christina Davy with us here today for a presentation. And I am Brad Baker with the Ontario Bat Network. For those of you who haven't heard of us, the Ontario Bat Network is a relatively new organization formed by a small group of individuals attending the second Ontario Bat meeting hosted by Toronto Zoo in 2019. Our simple goal at that time was to make sure that we could have a third Ontario Bat meeting, and we all see how well that's worked. Despite our best efforts, we were thwarted by the COVIDs. So we improvised, and here we are now with our speaker series, which has pretty much the same goal as the bat meetings, which is to get current research out to the broader Ontario bat community. Today's presenter is Dr. Christina Davy. I'm sure many of you have crossed paths with her at some point because she is definitely an active individual when it comes to wildlife science in Ontario. Christina is an adjunct professor with Trent University and works for the Ministry of Natural Resources and Forestry, and is a sitting member of the Committee of the Status of Endangered Wildlife in Canada, or COSEWIC. I think I speak for everyone when I say we're excited to hear what you have to say this morning. And with that, I will pass the floor to you, Christina.
Thank you, Brad. So can you hear me clearly? Sounds okay? Somebody, anybody?
You're good to
go.
Yeah,
great. Thank you. Okay. So thank you for the invitation. I'm really glad to be here this morning and share some of our work with you. And I want to begin by respectfully acknowledging that I'm joining you today from the treaty and traditional territories of the Mississauga Anishinaabe. And my team works on the traditional territories of a number of First Nations, and the species that we work with persist here today for us to work with them, in part because the millennia of effective care for the land by Indigenous people. And that allowed those species to thrive, and that work continues to this day. So I'm grateful that we also get the opportunity to chip in and contribute in a small part to the conservation of species at risk on those lands. I currently run a conservation ecology lab at Trent University, and I conduct research on species at risk for the Ontario Ministry of Natural Resources and Forestry. Fun fact, as of September 21, my research lab is moving to the Department of Biology at Carleton University. And MNR will be looking for a new species at risk wildlife scientist for anyone who knows a good candidate to keep their eyes out for the position. My research program explores the impacts of really rapid environmental changes, such as habitat fragmentation and wildlife disease or invasive species, on really small or declining wildlife populations. And we do this work in a variety of taxa, and it's all collaborative. So none of the work I'm presenting is just me. We have wonderful collaborators, and we have an amazing team of students and postdocs and staff. And so everything that I'm sharing was a team effort. And today I want to focus on some of our work with bats, because this is a bat group. And I'll use the time today to tell you about the work that we've been doing to understand the effects of white nose syndrome on bats in Ontario. And then more importantly, we've been exploring how we can best support the recovery of bat populations that have been impacted by white nose syndrome, now that that's no longer the key current threat to those populations. So this story is going to start in disease ecology land, but it won't end there. Really quick recap for anybody who's watching and is not familiar yet. White nose syndrome was first observed in 2006 in a cave in New York State, and it's caused by a fungus called Pseudogymnoascus destructans, which is endemic to Europe and Asia, and then was introduced to North America in or just before 2006. So the disease is named obviously for this fuzzy white growth that you see on the noses of bats that have really severe cases. And the species name destructans refers to its impact on bat populations. And this fungus is cold loving, so it only grows on the bats during hibernation, when they slow everything down and they drop their body temperature to that of the surrounding rock. The bats can carry viable fungal spores with them in the summer when they're active and they're bombing across the landscape, but those spores don't grow on active warm bats. They wait until the bats go back into hibernation, and the fungus can persist in these hibernation sites through the summer when there are few or no bats there. You've all seen the rainbow map, right? This is the spread of the fungus across the landscape from this point where it started, or where it was at least first identified. You can see that as the colors shift, you're moving through the different years from 2006 to more recently. This is also a very outdated map. It's moved farther west and a little farther north. But you can see that the spread is really rapid, and that's because the bats have effectively spread it among swarming sites and hibernation sites themselves. So most of this is not human-mediated spread. Little brown bats that get really severe cases of white nose syndrome end up with the fungal growth on the nose that gives the disease its name, but also all over the wings and the ears And this causes these lesions where the fungus actually invades the epidermis, and those resulting lesions disrupt homeostasis during hibernation. And there's this cascade of physiological effects that leads to dehydration and starvation and sometimes also mortality. So that's the bad news part. But it's important to know that not all bat species are affected equally by white nose syndrome. So I was initially really interested in understanding why this is. Is there something different about the response of susceptible and tolerant or resistant species? Or can susceptible populations adapt to become resistant? So together with Mike Donaldson, Craig Willis, and Chris Kyle, I designed a series of studies using gene expression analyses, also called transcriptomics, to explore how different bat species respond to the fungus and how the fungus responds to different bat species. And I'm not going to discuss those in detail here, but what I want you to understand is that the European bats, the ones that co-evolved with the fungus, are tolerant or resistant to white nose syndrome. So they can get it, the fungus grows on them, but the clinical signs or the symptoms are really, really mild. And it doesn't typically cause severe disease, and it doesn't cause mortality. Some North American bats, like big brown bats, seem to be more tolerant of infection, even though they didn't co-evolve with the pathogen. So we've had this really interesting time exploring the molecular responses of the different bats to the fungus and the fungus to the different bats. And the take-home message from these 4 studies, which has shaved years off of my life, is that the immune response of the bats differs, but more closely related species have more similar responses regardless of their evolutionary history with the pathogen. So the myotis that lives in Europe, the greater mouse-eared bat, picture like a giant little brown bat, has actually a very similar immune response to the fungus compared to our susceptible little brown bat. Whereas the big brown bat that lives over here in North America with the little brown bat, the big brown bat is more resistant to the fungus and has a completely different immune response. So it responds in very localized places on its skin where the fungus is invading, whereas the little brown bat and that greater mouse-eared bat mount a full system response to the infection. So we've got all this really interesting, gnarly interspecies differences. But moving back to the conservation implications of white nose syndrome, if we take a closer look at our susceptible little brown bats, they were hit really hard when the disease first arrived. And so in naive populations of little brown bats, you can see over 90 and sometimes over 95% mortality. So this photo shows piles of bones left on the floor of a hibernation site after white nose syndrome swept in. And this is one of the most dramatic selective sweeps that's ever been documented in real time. And it's obviously bad news, but our research is focusing on the remaining 5%. So maybe our little brown bat populations have the same genetic capacity to adapt to the fungus as those European bats that they're related to. Maybe adaptation was already happening. Maybe it wasn't as bad as we were afraid of. I wanted to know if 95% of a population of little brown bats dies from white nose syndrome, but 5% survive. What's actually different about the survivors? So are they just lucky so far, but they're likely to die of white nose syndrome soon? Or are they actually able to tolerate or resist the infection, like those species in Europe that co-evolved with the fungus? And this obviously isn't just an academic question. This is the information we need to design recovery plans that will actually work. And here's why. Let's say you have a population of bats and they have different immune gene variation among individuals. So let's say each color of bats here represents a different immunogenetic profile that's produced by previous selective pressures on the population, because there's always going to be other pathogens at play, right? You have a few options. Option 1, your novel pathogen comes in and causes 95% mortality. And the first scenario is that the disease is not exerting immunogenic selection. In this case, if you sample the bats before the pathogen came through and you compare it to a sample of bats from after the pathogen came through, so on the left of your screen here, you'll see that your sample will look the same. We have the same diversity of immune gene variants in the post-white nose sample compared to the pre-white nose sample. And this would tell us that the adaptation to the pathogen is unlikely. In scenario 2, the disease exerts immunogenic selection. And when we compare the diversity in the pre-white nose sample at the top to the diversity in the post-white nose sample, we see that particular immune variants are more likely to still be surviving, or the bats with those variants are more likely to still be surviving. And so that tells us adaptation to the pathogen is possible. And this matters because it helps us direct our recovery actions. In the first scenario, we would focus on treating the disease, right? We would focus on trying to eradicate the pathogen or finding effective treatments for white nose syndrome in infected bats. But in the second scenario, we would want to focus on supporting recruitment to this population. Spending a whole lot of energy and resources on treatments might not be effective if those bats actually can tolerate the disease. But in this case, we're now in small population land, right? Where we just need to boost recruitment and help the population grow again. So we conducted a sequence capture assay that was led by Mike Donaldson, targeting 138 immune genes and their regulatory regions. And we compared the immunogenetic profiles of little brown bats sampled from Manitoba all the way east to Prince Edward Island, and we sampled them before and after the arrival of white nose syndrome. And I'm not going to go into genetic details because I think everyone's more interested in the actual bats, but the take-home message here, and what you can see in the figure from this paper, is that we actually do see a signal of selection in bats that have been exposed to white nose syndrome and have not been exposed to white nose syndrome. And this was the first evidence for direct genetic selection by white nose syndrome on little brown bats. And it's great news because, again, this is what we would need to see. What am I saying? That's the wrong way to say it. Sorry. It's great news because it tells us that this population actually can adapt to tolerate the disease, assuming that we can support them while they're doing it. So we're doing some follow-up work on this at the moment, and Karen Vanderwolf, who's working on a PhD at Trent at the moment, is exploring how skin chemistry is linked to white nose syndrome susceptibility. And she's just finished this study characterizing pH variation in bat skin for the first time. This is in collaboration with Paul's lab at McMaster and with Chris Kyle at Trent. And so she's exploring how this pH variation affects the fungal and bacterial communities that grow on bat wings, and trying to understand how skin chemistry might vary between tolerant species and susceptible species, and how it might vary between little brown bats that have already gone through that bottleneck of being introduced to white nose syndrome and the ones that are still naive and have not yet gone through that. And our hypothesis here that little brown bats are adapting to white nose syndrome is also supported by our field data. So if the 5% of bats that survived were just bats that hadn't died yet, but they were less susceptible, then we predict an ongoing decline in the numbers of hibernating bats. And I'm excited to say that that's not what we're seeing in Ontario. So Lauren Hooton, who's also doing a PhD at Trent, is leading this study using winter bat count data from hibernation sites in Ontario caves and abandoned mines. So the x-axis here shows you the number of hibernating bats that were counted each year, and the counts indicate the total number of bats in each site each year. You can see there's that initial drop, right, where white nose syndrome comes through. We get that approximately 95% decline in the number of hibernating bats. And that's obviously horrifying, but notice that the two sites with the most bats, Craigmont and the Hunt Mine, which are both within 2 hours of Ottawa, showed a rapid decline after white nose arrived. And then they appeared to stabilize, with the numbers even increasing in recent years. So again, this is good news. This is telling us that recovery is possible. But of course, white nose syndrome is not the only infection these bats have to deal with. So Sonu Subudi and Vikram Misra recently led this study describing a novel native coronavirus isolated from little brown bats. The virus doesn't appear to cause clinical signs in the bats, but many of them carry it. So in presentations before 2020, I used to have to explain why we should study coronaviruses in wildlife, and I'm going to assume everyone is now sold on why this is important. Anyways, this particular coronavirus, which is native to Canadian and presumably also bats in the US, it has not ever been documented infecting humans. This is probably not a health risk to humans. It's in a different group of coronaviruses than the one that causes COVID. But as with all wildlife coronaviruses, there's obviously a potential for spillover, which is why Vikram studies them. And I came into this work because Vikram's team realized that this coronavirus was present in some bats that they had used in a previous experimental study of white nose syndrome. And they had stored samples. So Vikram said, well, what if we use the samples to build these post-hoc experimental groups of infected, uninfected, and co-infected bats? And then we use gene expression analyses, like the ones that I described before, to understand what happens when bats with the coronavirus also get white nose syndrome. So that's what we did. And again, very briefly, we had a control group where, so this is our pink group here, which are bats that didn't have the coronavirus and were not infected with the fungus that causes white nose syndrome. We had a group shown in blue here, which did not have the virus, but were infected with the white nose fungus. We had a co-infected group that had both pathogens, and then we had a virus-only group shown here in yellow. And what we found is that the co-infected bats developed more severe clinical signs of white nose syndrome than the bats that only had white nose syndrome. We also found that when the bats upregulated their immune response to the fungus that causes white nose syndrome, they ended up downregulating their antiviral responses, and we saw a 60-fold increase in viral shedding. So this dynamic has obvious implications for spillover risk, right? Bats with white nose syndrome are shedding more coronavirus than bats without. And there's a potential link to rabies shedding here as well, although that's something that needs more research. So there's lots of work to do on that disease ecology front to understand more pressures on little brown bats, and we're still working on that. And we're involved with some coronavirus screening with Samira Mubareka's team at Sunnybrook Health Sciences to try and get a better idea of the diversity that's out there and screen for potential COVID spillover back into bats. But if we come back to recovery of bats that we're dealing with white nose syndrome, and we try and take all this academic information and turn it into useful suggestions for informing policy and species recovery plans, we know that the bats here seem to be tolerating white nose, and that this tolerance or resistance is apparently heritable, right? So this means that the recovery actions for those bats should consider the impacts of co-infection and should focus on maximizing survivorship in affected populations rather than focusing on the disease itself. Very quickly, I just want to remind you of something everybody here knows already. Bats are not mice. They reproduce very, very slowly, and they have these giant babies. And the energetic requirements for a bat to raise a pup successfully in the summer are huge. So if we think about bats that are potentially tolerant or resistant to white nose syndrome, they survive through hibernation, and then they emerge. Then the females have to pick up enough energetic resources to birth gigantobaby and then nurse gigantobaby through the summer. And that's huge, even without the extra pressure of surviving white nose syndrome first. So with some awesome collaborators, and this included Craig Willis's lab and Gabby Mastromonaco at the Toronto Zoo, we asked whether the impacts of surviving white nose syndrome carried over into the summer and into this really energetically intense reproductive period. One way to measure physiological health is that we can quantify chronic stress. And chronic stress is like basically the stress that you experience over a long period of time, and that stress is mediated by stress hormones such as cortisol that your body produces in response to either stressful events or also just on a daily cycle that helps you remember to eat when you should be hungry and so on. Stress hormones accumulate over time in your keratinized tissue. So you have a record of your stress over time in your hair, for example, or in your fingernails. And it also accumulates in bats' claws. And because the deposition of tissue into the claws slows down during hibernation, the claws of the bat at the end of hibernation can tell us about the stress it experienced the previous summer. So we did a whole bunch of postmortem pedicures of bats and tried to understand whether they were experiencing more stress if they had white nose syndrome the previous winter than if they didn't. So if you look at the cartoon here, you've got sort of the yearly cycle of a bat. They're hibernating in the winter, they emerge in the spring. If they had to also deal with white nose syndrome over the winter and they survive, they now have to stick this costly recovery period in the spring. And the photos here are showing you examples of bats that emerge and then mount such a strong autoimmune response that they dissolve their own wings in doing so. So the recovery can be really costly as well. Then they go into reproduction, then they're finally swarming in the autumn, and then they go back into hibernation. And again, this is already really constrained even without white nose syndrome. When you add it in, it becomes more constrained. So those claw samples that we collected in bats that had experienced at least one winter with white nose syndrome are telling us about cortisol deposition, stress hormone deposition, the previous summer. And we found that, yes, bats that had gone through at least one winter dealing with white nose syndrome did experience much higher stress hormone levels in their claws, as documented in their claws, than bats that had not yet been exposed to white nose syndrome. So this is the first documented example of a carryover effect from a pathogen. That means an effect that lasts beyond the immediate impact of the disease. And again, this is telling us that these bats really need strong support in the summer to deal with the impacts they experience from the white nose syndrome that they may have during the winter. Okay, so let's say you're a little brown bat that survived white nose syndrome. You can tolerate the disease. It is possible that part of the selective process for these tolerant or resistant individuals also includes that they don't experience such a strong stress response after recovery. So let's say you come out and otherwise everything's great. You got some white nose syndrome in the winter, but it was okay, you can tolerate it. You're not mounting a massive stress response now, you're okay. So you come out, you still need to find a safe roost where you can produce your giant pup. And you still need foraging habitat that is safe and filled with lots of tasty insects. So there's this direct link between protecting and recovering bats and protecting wild habitats that support healthy and diverse insect communities, like the wetland that's shown here. And of course, here's the challenge. The bat's roost site is defined as habitat, and it receives protection under our existing policies. But the foraging habitat, the air where the bat spends all of its night feeding, and the air that it has to fly through to get from its summer roost to its swarming site, and among swarming sites, that aerial habitat is not clearly defined and therefore it's not protected. And of course, there are many threats to aerial species that are not on the ground or in the water, and often wildlife is killed while flying. That can include collisions with wind turbines like these, or with cars or buildings or airplanes. But again, aerial habitats aren't explicitly recognized in existing policy. And this isn't a problem that's specific to Canada, this is worldwide. So even the Barron Convention on Migratory Species doesn't explicitly recognize the airspace as habitat. So a few years ago, I teamed up with some colleagues to try and think about solutions, and we tried to come up with some examples of classifications for aerial habitat that matched the layered classifications that are used for ocean habitats, which are classified by depth. So in this case, we classified by altitude above ground. And this is just a cartoon showing you some examples of species that are found flying at various heights. But the point is, even with two bats here, so the nocto, which is a European species, and then we've got an example of a red bat farther down. You can see that these species are flying quite high, and that if we're not protecting that airspace that they're using for flight, we may not be fully protecting them. And if you're feeling like, well, I mean, they could just fly over top of things, they can, but then they run into all sorts of challenges as well. So we made this really rough illustration just to try and get our point across. So what you're seeing here is a migratory pathway that, say, a hoary bat might take if it was flying from sort of Florida north to our area. And once it starts, what you're seeing are all the different things that intrude into the airspace from a 0 to 1 kilometer altitude. And what I want you to notice here is that if you were flying 1 kilometer or lower, it would be very difficult to fly in a straight line, right? You have to do a lot of navigation back and forth. The other thing I guess you should probably notice, I'm just going to play this one more time. The colors are showing you in red airports, blue cities, black roads, white wind farms, that's the part I want you to focus on, and purple is FCC communication towers. And I want you to notice how the proportion of wind farms really increases as we get farther north in the northeastern part of North America. So the white parts coming up, those are the wind farms there. And if you were trying to fly up the shore of Lake Michigan, for example, you could either fly out over open water, which not all species want to do, or you would have to navigate all of that really complex fragmentation. So we're really trying to dig into this right now, and we're doing that in a few ways that I'm really excited about. So this is Dylan, who's studying 3D aerial habitat use of hoary bats using little tiny altimeter GPS trackers. And he's trying to understand how bats are using that habitat in a 3D way, right? Instead of just the point on the landscape map, how high they're going and how that affects their collision risk with things like wind turbines. We've also done some studies looking at how bats in Ontario are affected by wind turbines, and I won't spend a whole lot of time on this, but there's a point I want to make here that comes back to white nose syndrome. So we took data from all of the facilities that had contributed data to the Government of Ontario as part of their agreements to produce wind energy, so 48 facilities, 594 turbines. And we found that the effective captures of bats by turbines decreased over a 7-year period quite substantially. And the decrease ranged from 91% over 7 years in silver-haired bats to 65% in big brown bats. And that big brown bat slope looks like it's maybe no big deal, but it is a 65% decrease in the number caught in the airspace. And the reason I want to flag big brown bats here is because I often hear people say that they're doing fine. If we only had big brown bats on this figure, right? If we didn't have hoary bats or red bats or silver-haired bats in Ontario, and we had a 65% decline in big brown bat apparent abundance in the airspace, we would be freaking out. So I think I just really wanted to encourage everybody to watch out for that shifting baseline that we have sometimes, because I know that hoary bats are obviously in more trouble, but big brown bats have the double whammy of mortality at wind farms and potentially from white nose syndrome, which they do get. And I think they need us to pay a lot more attention to them. And then in a more landscape scale way, we're also just trying to understand how bats are moving across the landscape. And we're doing this not only using the GPS tags that Dylan's putting on hoary bats, but also building this big modus network around a whole bunch of swarming sites. So anyone here who's worked with Brock Fenton and comes from Brock's long bat family tree, which I think is many people here, we're basically building on the swarming work that Brock started doing in the 60s and that everybody on his team has been continuing since then. And we're building a modus network around these swarming sites to try and understand regional migrations and better understand how bats are moving between the sites. We're hoping that this will complement some of the other great work that's going on. So you probably all know Craig Willis's lab has this amazing network of pit tag readers that can understand bat movements into different swarming sites and the timing of those. And we didn't want to duplicate that. And Hugh Broder's work with movement between maternity colonies, again, like it's so far ahead of anything we could possibly catch up to, and we didn't want to duplicate effort. But what we're really curious about is the pathways the bats are taking between these sites. And so we're hoping that adding this into the mix can help complement those other long-term projects and give us a better idea of how bats are using aerial habitat and, again, how we can best protect them. So I just really quickly want to acknowledge all the funders that have helped us to do this work. And again, I did not do this alone. I am so lucky to work with the students and postdocs and other collaborators that I get to work with. And I'm really grateful I get to do this. And I want to thank you for your attention as well. And I'm happy to answer any questions that you have.
Excellent. Thank you, Christina. Wow, quite an interesting story. From white nose through coronavirus to wind turbines, hunting insects is not easy. Thank you so much for your presentation, and I look forward to seeing what comes out of your lab as you move to Ottawa.